But it's with great pleasure and honor to introduce our next speaker for our IASIC session, Dr. Yasmin Hurd, unlocking the biological impact of developmental cannabis and its psychiatric risks. I'm sure we're in for a very good presentation today. Dr. Yasmin Hurd is a Ward Coleman Chair in Translational Neuroscience, as well as Professor of Psychiatry, Neuroscience, and Pharmacologic Sciences at the Icahn School of Medicine, New York. She's also the director of the Addiction Institute at the Mount Sinai Behavioral Health System. She is an internationally renowned neuroscientist whose preclinical and clinical translational research examines the neurobiology of substance use disorders and related psychiatric disorders with primary focus on opioid abuse and the developmental effects of cannabis. Based on her high scientific accomplishments, she was inducted into both the National Academy of Sciences and the National Academy of Medicine. We're very happy to have Dr. Hurd share us her knowledge. Just a couple of housekeeping issues. I'm going to mute myself. I'll let her have at it with her slides. If you have any questions, please put them in the Q&A box, not necessarily the chat, and we will try to address them after she's done with her presentation. So, Dr. Hurd, thank you so much, and I'll let you go for it.
Thank you so much for the invitation. I mean, this is a really important group and all the things that you're doing. Let me share my slides. So what's that? Okay, hopefully you still see all the slides, and I'm going to take care of all of these floating things around. But as I said, thanks again for the invitation to talk to you today about clearly one of my, I would say, passion for our society in terms of understanding the developmental effects of cannabis. And I don't have to say anything for, especially this group, in terms of this pendulum that we've been, going through for many, many decades, even centuries, in terms of whether the cannabis as a recreational substance that, people thinking there's no harm, it's healthy, it's not addictive, it helps treat their anxiety. Basically, it's a wonder drug. To the other side of the pendulum, where, we talk about the psychopathology associated with cannabis, including, psychosis, negative affect, in fact, that it may induce anxiety, and of course, the addiction risk. We know a lot more about cannabis than we've ever done before. Clearly, this is a very complex plant. We know that it contains hundreds of cannabinoids. Most of them we still have no clue about. And a number of them actually have this psychopharmacological effects in the body, as said, that we don't know much about. And not only the cannabinoids, but all the other chemicals and the terpenes as well. It's clear that this complex plant has changed over the years. And for example, the concentration of THC, the main psychoactive cannabinoid in the plant that's associated with the high, and also associated with more of the negative effects that we see in relation to psychiatric risk, as compared to another cannabinoid like CBD that we'll talk about later, where the ratio of THC to CBD being considered more, well, one, it's non-intoxicating, and it has more quote unquote beneficial effects that we'll see with research. And here we know that THC over the years has increased a lot as compared to CBD. But it's not just only the over 500 chemicals. It is the historic high of the potency of these THC products today, and the very route of administrations that are very different than in previous decades, even the past 5 years. And we now have the chemical synthesis of new type forms of cannabinoid products. For example, CBD, that is a non-intoxicating cannabinoid, as I said. Now they're chemically converting it to THC analogs like these Delta 8 products, Delta 10, that have psychoactive effects. And even rare cannabinoids that you needed this, high-tech machine in order to even detect it in the normal cannabis plant. Now they're making it synthetically. And some of these cannabinoids are even 30 times greater than THC. So this is why we see that cannabis use is increasing over the years in the US. And in fact, the near daily and daily use of cannabis is now higher than the near daily use of alcohol. And I don't think that anybody ever expected that would ever happen in our society. So the complex plant, the complexity of the cannabis, I would say, industry that created all these products, but there's also the aspect of the endogenous biology of our natural endocannabinoid system that is also complex. And many people have said, we talk about THC, the plant-derived cannabinoid that is associated with all of these, the effects that we know in regard to intoxication and so on. It binds to cannabinoid receptors, and there are mainly two cannabinoid receptors in the body called CB1 and CB2. They're expressed here in this schematic cartoon on the left. You can see that they are expressed throughout the brain and body. The brain has mainly the CB1 receptor that I'll come back to, but basically every organ, including our skin, contains cannabinoid receptors. And they're not there for THC. We know that they're there to mediate the actions of our endogenous cannabinoids, these endocannabinoids. And there are two main endocannabinoids that have been studied in the field, but we know there's much more than that. And these endocannabinoids are really, we say, synthesized on demand. They're synthesized from lipids, and they're relatively recent. So many people think that because cannabis has been around for thousands of years, we know everything about the cannabinoid system, and we don't. Our endogenous cannabinoid system, we're learning a lot about. In fact, if you look at these two endocannabinoids, that mainly those are the main two that the research field focuses on, they're actually part of this huge lipid system. And most of these lipids, we, again, know very little about. So we're talking about complex plant and complex biology. Again, we're still learning a lot. But the one thing that we do know that the endocannabinoid system is a key modulator of a number of biological functions. As I said, we know of the two main cannabinoid receptors, CB1, CB2, but there are many other cannabinoid receptors that are at least receptors that can mediate the actions of endocannabinoids throughout the body, in addition to obviously the endocannabinoids themselves, AEA and 2-AG that I mentioned earlier, and the enzymes that synthesize them and degrade them. They are critical for modulating many biological functions, including obviously energy metabolism, appetite regulating, our thermostat, immune function, neural development, pain and nociception, sleep, stress, and even reproduction. So they are critical. And this is one of the things that I think that many people think, cannabis is this benign drug, but it's this quote unquote benign drug impacts on large biological systems in our body. I'm a neuroscientist, so for me, understanding the endocannabinoid system was one of the things I wanted to look at early in my career when I started being interested in studying the effects of cannabis on the brain. And these sections here are images of the human brain that's showing the expression of the cannabinoid, the CB1 receptor. And one of the things that we could see is that it's highly abundant throughout the brain. In fact, the CB1 receptor is the most abundant receptor sitting on the surface of cells, the most abundant. We see it throughout the brain. And it's not only that it's widely abundant throughout many brain areas, but it actually where these receptors sit, they sit at the synapse regulating the communication between cells. So they play a very critical role. My question, and that obviously of many people, and why some of you, come in today to hear this lecture, is the relevance of cannabis and psychiatric risk. Of course, cannabis use disorder is a big question that we all have, especially with the increasing THC concentration. And in fact, many people may think that you can't develop a cannabis use disorder, but approximately about 30% of people who use cannabis do develop a cannabis use disorder. And that's similar to other substances of abuse like opiates or so on. But it's not because cannabis has as high addictive potential as opioids and also stimulants like cocaine. But as the concentration of THC is increased, so has its potency, and we see greater increase in cannabis use disorder. But it's also been linked to a number of other psychiatric disorders, including the psychosis, schizophrenia, anxiety, and depression. And there's a high psychiatric comorbidity with cannabis use and a number of I said, of these psychiatric disorders. Now, the question has, been why? And for me and for a number of people, we now realize that a lot of psychiatric disorders, although they're diagnosed in adulthood, they have a developmental origin. And we know that adolescence is a time period where a lot of people use drugs, and adolescence is a time when you have a higher I should say the initiation of a lot of the symptomatology that predicts certain psychiatric disorders later in life start to emerge. But even prenatal exposure to drugs and prenatal environments can also lead to long-term impact on adult psychiatric risk. And the reason why early exposure to cannabis is particularly important in terms of psychiatry is that the endogenous cannabinoid system plays a really critical role in many neurodevelopmental processes from the beginning of life to the development of cells and neurons, to how they migrate, the synapses that they form, and our endocannabinoid system is really critical at all of these stages, even into adult adolescence, when they are critical. For some reason, my slides are going backwards here, but they're critical for even the maturation of the prefrontal cortex, that last part of the brain to achieve full maturation. And the endocannabinoid system are very dynamic during all of these developmental processes. And as I said, we know that now more women who are young women of childbearing age and during pregnancy, more people use cannabis, even during breastfeeding. And with the legalization of cannabis throughout most states in the US, there's an increase in cannabis indoors and for kids. And of course, teenage is a time when many people used to experiment with drugs. So I want to start with, what do we know about the what do we know about, I should say, the developmental effects of cannabis? And a large part of it does come with understanding the endocannabinoid system. And as I said, when we look at the cannabinoid receptor, I showed you before the image of the adult human brain. Here is the fetal brain. And we could see that the cannabinoid receptor is highly expressed. But actually, what's interesting, it's highly expressed in these brain areas that we call the limbic related areas that are really critical for emotional regulation. But does it really have an impact in utero exposure to the human brain? And one of the things that we could see from very early on was that in the fetal brain, those that had been exposed to cannabis in utero showed changes in what we call the cytoskeletal, the actual structure of the brain. And that's actually fitting with the fact that we know that the endocannabinoid system is critical for hardwiring of the brain during early development. We could see it on multiple transmitter systems here, on even opioid peptides. And these peptides play a really critical role in reward, motivation, even stress and pain. And we could see this in the brain, and it even correlated to the amount of cannabis, the alterations on these opioid peptides that the mothers had reported using during pregnancy. We even saw it in transmitters that many of us in the addiction field have focused on a lot, dopamine. And, dopamine is critical for mediating reward, learning, attention, motor function. And we could see that this dopamine, for example, D2 receptor, is reduced often as a characteristic feature that we see in adults with substance use disorders. But here we saw it in the fetuses that had been exposed to cannabis. And even this is a brain area called the nucleus accumbens, again, important for reward, and another brain area, the amygdala, which actually expresses a lot of the cannabinoid receptors in the fetal development. And we could see even here that males showed a much greater effect on this dopaminergic system. But when you study humans, it's always challenging because are those findings specific to cannabis? And the question is there a long-term effect on adult brain and behavior? And so that's when we, use our animal models, and we could actually replicate what we saw in the human fetuses. So for example, again, in the area of the dopamine, just showing one, for example, the dopamine D2 receptor that was reduced here in this nucleus accumbens. And when they got older, it was still altered. And the only difference was that they had prenatal exposure to THC. We could also see that the firing of their cells here, this was in the area called the hippocampus, differed in adults that had been exposed to THC during prenatal development. But the question is, those are animal models. Does it relate anything to human? And I'm a masochist, but my colleague Yoko Nomura was even more of a masochist than I am, because doing developmental studies are not easy. And she was interested in looking at stress during pregnancy. And I wanted to look at, perhaps other substances like cannabis as well. But many pregnant women will have experienced different types of stressors. But we had in New York at that time, in the Northeast actually, when we started, a little bit after we started this study, Superstorm Sandy, the hurricane that basically, half of New York here, Manhattan here, you can see was underwater. And so in studying women before, during, and after Sandy, we wanted to look at, is there a long-term effect on not only on the women, but really on their offspring? And so to get an idea about the in utero environment that their offspring experience, we looked at the placenta. And the placenta obviously is the most critical temporary organ that exists. It gives life. It's important for supporting fetal development. But we also know that placental programming is actually essential for not just neurodevelopment, but it's linked to psychiatric risk later in life. So in sequencing the placenta, the question was, does cannabis impact the placental environment? And in doing that, at the time, my postdoc, Greg Rampala, we could see that there are many genes. When we sequenced to look at thousands of genes in the placenta, we could see that many things were changed, including the cannabinoid receptor gene, and it correlated even to the amount of cannabis the moms reported using. But the thing that was changed in the placenta, to our surprise, very robustly, were these immune-related changes. And no matter how we looked at it, these came out. Other members of my team later on, other postdocs, have replicated this. And here, this is us looking at genes that are increased or decreased in the placenta, and the yellow ones are those highlighted, are those related to the immune system. Again, we see these really strong changes in the immune system. But not only in the human placenta that we see these, we see that the males have the most predominant changes in the immune changes compared to the fetal, the female placenta. And we could replicate this in our animal model, that they were given THC, vape THC, and we could see once again that immune-related changes were there in the placenta, and again, predominantly in the males as compared to the females. We could also look at the proteins, not just the genes, but the proteins. And once again, yellow emphasizing really strong changes in the immune-related proteins. So when we look at what the changes are, we could see that the biological processes, as I said, many of these changes related to immune, I'm just highlighting immune, and also mitochondrial function, that powerhouse of the cell that really is critical for regulating biological processes, and even the genes that are regulating biological systems that regulate those immune function, that are really important for anti-inflammatory and mitochondrial oxidative stress processes. I know perhaps I'm saying certain words that may be too detailed, but the take-home message that not only the human placenta, but also the rat model replicated exactly what we saw in the human. And again, these immune-related changes, these mitochondrial ATP-related, oxidative stress-related changes in the placenta as a consequence of maternal cannabis use. I want to add just one quick thing, just because many people may know of more novel, or I should say, know of classical neurotransmitters. It's not only immune-related changes that we saw. For example, when we looked at the thousands of proteins, we could see that there are some proteins that were not expressed at all in the placenta of the animals, and others that were. And one of them I'm bringing attention to mainly because this particular enzyme, it actually is critical for degrading tryptophan, which is the precursor of serotonin, a classic transmitter system that's highly implicated in psychiatric disorders and also even in immune function. And we saw that not only was it changed in the placenta, but even serotonin levels were changed, and primarily in the males. So once again, there is a sex-specific effect that we see with prenatal cannabis and prenatal THC in our animal models. So a question is, does it really matter what happens in the in utero environment to the offspring behavior and their traits later on in life? And so my colleague Yoko Nomura, children come back every year or so and go through a battery of neurobehavioral tasks, just showing you. One of the things we also measured was stress hormone. This is measured in hair, and we could see here the kids, they're 3, 4. Those in the pink here are those that had been exposed to cannabis in utero, and they show much greater stress hormone levels. They also show greater clinical behavioral changes in regard to aggression, anxiety, and hyperactivity. And it's important to emphasize it wasn't just the cannabis. We saw that there was this synergistic interaction with those moms that had also experienced a lot of stress during pregnancy, that even the anxiety was, off the charts in the kids with that way. And even they showed an earlier diagnosis in those kids that had both the stress experience and cannabis exposure. So it's not just cannabis on itself, but the environment is also important as well for that. Importantly, one of the reasons that we also did this study was to try to see, does the early life environment makes a difference in predicting behavior later in life? And we could see that the gene networks in the placenta that related to anxiety, to immune function, actually correlated to anxiety only in those kids that later in life that had maternal cannabis use. So we've replicated again, and we can see again that the immune-related changes related that we see in placenta relate to anxiety later behavior in their kids. But also we see that these genes related to the lipid processing, you remember totally about the endocannabinoid system, actually relate to their depression. We also can look at our animal models to see whether or not there are effects long-term on behavior. And, given the fact that we saw these changes with the lipids, just showing that we saw that the placenta did have changes in the endogenous endocannabinoids in those that had been exposed to THC and some CBD. I'm going to go past that. So the question was, do you see this in animal models? And the one thing that the study in the children told us was that often in our animal models, when we had looked at prenatal THC or cannabis exposure, we were looking in adulthood. But the human studies told us that you can see changes from very early on, even during infancy. So we can see changes in affective behavior, and I'll go through some of this as well. Here's some of the data that the first signs in animal models that you can look at is how the pups communicate with the mom, for example. And we could see that there were significantly big differences in their communication in the pups that have been exposed to prenatal THC. We also saw that their circulating levels of their immune cytokines also differed. And it differed in the sense that it was males that showed a greater change in their circulating immune system. When they're adolescents, we saw once again that there were significant changes primarily in the males that have been exposed to THC during during pregnancy, and not much change on the social plane. Again, when we saw anxiety-like behaviors change in both males and females, even though during adolescence, and we see that this behavior persists into adulthood. And we even see changes on immune circulating levels in those that have been exposed to THC. Once again, the males show a greater effect, at least for this particular immune mark. As I said, we see that these things last into adulthood, but even from my previous older studies, we had seen this. So adult animals that had prenatal THC exposure, and they can get exposed to opioids where they choose to self-administer opioids. And because we were looking at, is there opioid sensitivity for addiction risk? And we could see that these animals, adults that had been exposed to THC during prenatal life, they self-administered more heroin. But even when you adjusted the conditions so they would take the same amount of drug, those adults that had prenatal exposure to THC, they would be the first to run to push that lever to get heroin. And even when they had a mild stress, they would seek the drug out more and take more of the drug. We see other behaviors change in terms of their changes in their motivated behavior, depression-like behavior, and even in their reward hedonic state, that again, when they're stressed, that exacerbates those types of affective, emotional type of behavioral responses. So for me, a big question is, what are the biological mechanisms that are really maintaining these long-lasting effects across life, just as a consequence of, early life exposure to cannabis or THC? And that's where we started studying epigenetic mechanisms, because we know that this is not a genetic effect. It's environmental. And the environment can control gene activity through modulating these epigenetic mechanisms. So genes that should be closed and turned off can be opened by these environmental tags that change and push open, so that genes now that should be turned off, as I said, are now turned on, and vice versa. There are numerous epigenetic mechanisms. I'm not going to go into them, but one of them was particularly interesting for us because whenever we studied the different cohorts of our animal model with prenatal THC and looked at different epigenetic marks, this particular epigenetic mark kept on coming up as being altered by prenatal THC. And this is a histone methyltransferase, meaning that it's really a transcriptional activator, and it plays a really critical role in maintaining, I should say, like a environmental memory on the DNA. And we can see that this epigenetic mark lasts throughout life, actually. But importantly, in an animal model, we could go in and change the expression of this gene in the brain of the animals, and we could normalize their behavior. So we know that these epigenetic mechanisms that are changed by prenatal THC are causal, I should say, to prenatal THC and relates directly to the behavioral outcomes that we see in adulthood. We also saw that this epigenetic tag, it actually regulates many immune-related genes in the brains of these adults. So that on the right side here, this is the relationship of these epigenetic tags to immune genes. And you can see on the left, there's a complete reorganization in those adults that had THC exposure. So these epigenetic enrichment are keeping these tags way long into life, or way across life from prenatal exposure to THC. But what about adolescence? As I mentioned, adolescence is this very critical stage for what we see vulnerability to psychiatric risk. And a large part of that is due to, this is when the brain really fine tunes itself, where, cell communication improves and leads to the full maturation of the brain. And we know that, as I said, many teens, these numbers now, we know that more teens are using cannabis than before, at least high as a cannabis. But the important thing about adolescence is that we know that most adults with a substance use disorder, they initiate their use during adolescence. And adolescent cannabis use is the most critical window for developing a cannabis use disorder. And we know that many young people who use cannabis meet this diagnosis for cannabis use disorder. And this is one of the issues that we have to address. But it's not only cannabis use disorder. We also know from many studies of colleagues throughout the world that here, this is a study done in Denmark, where they looked at the relationship between cannabis use disorder and schizophrenia. And in their society that really didn't have that much drug, and especially even cannabis, as cannabis became more prominent in their society, they saw this increase not only in cannabis use disorder, but in the number of cases with schizophrenia. But there was a sex effect, so that this was much greater in males. And in fact, the preventable cases of schizophrenia that related to cannabis use disorder was as high as about 30%. So it's really critical in terms of schizophrenia risk, but it's important, and even cannabis use disorder, it's important to realize that people may not even meet a cannabis use disorder, but still show here, this is now this being called non-disorder cannabis use among adolescents, meaning they may not meet the clinical diagnosis of a cannabis use disorder, but they still consume significant cannabis and show this mental health, cognition, academic performance issues. And those are things that we don't need to wait until a child or adolescent develop a cannabis use diagnosis before realizing that there's an issue. And human neuroimaging studies have shown that there are changes in the cortex with adolescent cannabis use, and that this accelerated cortical thinning that even correlated with impulsivity in these young people. Human studies, again, there's always debates. So we go to our animal models and we can look to see whether or not does adolescent THC exposure change anything in the prefrontal cortex, in these beautiful cortical cells that are really critical for our cognition. And one of the things that we could see is that adults with adolescent THC exposure, the complexity of their cortical cells differed significantly. And the changes actually resemble what you see with stress. And we went and we picked up these cells with a laser, so with laser capture, and then we sequenced them to look at what was actually changing these cells. And at that time, I was very surprised because there was a complete reorganization of the gene expression signature in the cortex of these young adults as a consequence of adolescent THC exposure. In fact, they only shared about 5% of the changes for the normal developing adolescent brain. And where the changes were initially not surprising, we saw these structural changes in the cell, and that's what the genes showed, that the structure, the genes that are critical for cell projection and the development of these cells changed. But also these epigenetic changes. In fact, the same epigenetic mark that we saw with prenatal cannabis exposure was also changed in the cortex of these animals. And the correlation between these epigenetic genes and these structural related genes in normal animals, it's not dramatic, but there's a complete reorganization of that relationship in adults with adolescent THC exposure. And these changes mimicked what we saw in the prefrontal cortex of individuals who were diagnosed with schizophrenia. So there are some common biological mechanisms by which adolescent THC is changing the gene expression signature, similar to what we see in individuals that have psychiatric disorders. So what has changed over time as I started my talk is this aspect of potency, that THC over time has changed dramatically in not only the flower that's there now, like 24%, and some even more, some cannabis strains, but even in the delivery systems and the dabbing and these highly the concentrates that put THC levels even up to 90% of some of the delivery systems. And that's where we know that's the aspect of the pathological effects of cannabis. Our normal, as I said, endogenous cannabinoid system is critical for our life. It's important for regulating so many key biological systems and the normal synaptic function. But as it pushes with this high THC concentrations, it pushes us off to more pathological states. And we know that these high potency cannabis products does increase the risk of psychotic symptoms, depression, anxiety, and as I said earlier, cannabis use disorder. And people think that, teens will titrate their use of these high potency cannabis products. Adults don't even titrate that use, and that's the issue. So we can use animal models and look at, does dose matter? And when we look at the stress hormone in animals that were given what we consider, low dose THC and high dose THC as adolescents, their stress hormone levels last even months after their final cannabis exposure. And if you give them a minor, mild stress here, just acute isolation stress, they show greater lever pressing for rewards. And we can see in brain regions that are critical for regulating like decision making and anxiety that there are changes in the cannabinoid receptors in these particular cells, and that they correlate directly with impulsivity. We can also see aspects of decision making. Decision making is a critical cognitive facet in most psychopathologies, including addiction. And even in human cannabis users, they have this Iowa gambling task where they have cards, and these decks of cards are stacked for either being advantageous that they will win, or disadvantageous in that they may win initially big amounts, but they will ultimately lose. And cannabis users, individual adults with cannabis use disorder, always show this impairment, risky decision making on these tasks. We can also do this in animal models. Instead of money, they get, sucrose pellets, and they show with the high dose animals show the same thing as human cannabis use in individuals with cannabis use disorder, meaning that they show risky decision making on these gambling tasks. They also show that if they get exposed to THC again, these were adults that had adolescent THC exposure, that if they get exposed to THC again as adults, they again show increased risky decision making and greater impulsivity, and in all of the different measures that we looked at. Jackie Ferlin, when she was a postdoc, my team developed the model of looking at edible THC, because that's now what a lot of teens are using. And we could see that the self-administration in the amount of cannabis edibles that the animals consume, that those that consumed more THC as adolescents showed greater risk of decision-making, just like our human cannabis users. And when they're stressed, they consume more THC. So neurobiologically, we can look into all of this, and I can, talk to you more about all of the things that we see in the brain. But one of the things that we do know in that a lot of the brain areas that are critical for emotional regulation, that genes are changed dramatically in these areas with stress. But it's with combination with high dose THC, they're changed even more dramatically. And here, for example, there's particular cells that change a lot. They're called astrocytes, a type of glia that are actually important for even immune function. And we can see it's not just in the expression of these genes, but the actual structure of these cells change with high dose THC, and especially in regard to stress. And they correlate with the risky decision making and impulsivity that these animals show. So the endocannabinoid system is critical for regulating stress. In fact, it's our endogenous biological system that inhibits the stress response. But what happens is that our endogenous cannabinoids that are there to mediate the effects of stress, when you have frequent cannabis use, here colleagues at Yale that had looked at the brain, and you can see cannabinoid receptors are reduced in cannabis users, you see the same thing throughout the body. And so what happens is that the cannabinoid receptors with cannabis use, not only do they change the natural levels of our endocannabinoids, it changes the receptors that are there to be the brake on the stress response. And so therefore you get this poor response to stress with frequent cannabis use. But many cannabis users will tell us that cannabis helps them in alleviating their stress, their anxiety, but actually it does the opposite. So developmental cannabis exposure, potency does matter. Early use matters. The frequency of use matters. Biological sex does matter. We see a lot, at least in terms of some of the prenatal effects with males, but the gap is closing. We see a lot also in females. I didn't go through aspects of biological traits, and psychiatric comorbidity does matter. Stress matters a lot, and that's part of the environment. But the cannabinoid also matters. So I'm going to just end in the last part here with reminding you that the complexity of the cannabis plant with over 500 chemicals. In our animal models, we always focused on THC because it is the main psychoactive cannabinoid associated with the reward, the addiction, and the psychosis risk. But CBD, as I mentioned, is also a cannabinoid. Back then, when nobody was studying CBD, we wanted to just look to see what does another cannabinoid do. And this cannabinoid, we didn't know that much about it. We knew it was not intoxicating, at least seemingly so at that time. And when we looked at it in our animal models, we were very surprised. So if you look at adults that had adolescent THC exposure in green as compared to the control in blue, they would self-administer more heroin, just like we saw with our prenatal animals. And if you give them stress, they would self-administer even more. But when we gave animals CBD, it actually decreased their heroin seeking, meaning we usually, like in humans, when they get an addictive drug, they peer something in the environment with that cue with getting the drugs, whether it's their friends or environment. Here for our animals, we would make the light go on when they got heroin. And when we gave them CBD, it reduced that. We also, based on our animal work, we wanted to see could CBD potentially, in fact, reduce opioid addiction. And we did a short study looking at the same dosage that we gave our rats and look at individuals that had opioid use disorder and looked at, for example, craving. We showed them cues with videos. And when those who had received placebo saw the heroin cue, they craved, and CBD reduced that. And these were double-blinded, placebo-controlled. A week later, CBD was still reducing their craving. A week after their last CBD administration, then that's what we saw in our animal models, that weeks later it was still having a positive effect. But what our animal models at the time didn't tell us was anxiety. Our humans told us that they showed high anxiety when they saw the heroin cues, and CBD reduced that. A week later, it was still reducing that. We also looked at their cortisol level, and when the placebo individuals saw the drug cue, their cortisol stress hormone levels went up, and CBD reduced that. Similarly, with their heart rate went up when they saw the drug cue if they had been given placebo, but those that had given CBD, it reduced that. So now we go back to our animal models, and we can look at, again, does CBD decrease heroin-seeking behavior in our animals, cue-induced seeking, and it does. But we can also now look at anxiety-like behavior. And we can see that, interestingly, animals that have this shock, this trauma, and show anxiety-like behavior, when you show them that anxiety cue, those that have controls, basically the placebo, they show high anxiety-like behavior, and CBD reduces that. So replicating what we see in our humans. We are also looking at different lipids and endocannabinoid systems, and we can see that their CBD actually normalizes a number of the biological processes that I saw, even that we saw with the placental changes, we see in the brain of these mitochondrial and immune function, that CBD actually reverses it, and it correlates with improving their anxiety-like behavior. But there are common biological mechanisms that we see that CBD impacts on in normalizing, whether it's in cue-induced heroin seeking or in anxiety that we're following up on now. So cannabis, it's really critical that perhaps we stop calling it cannabis, but I know it's difficult in in the real world, but it's really about the cannabinoid exposure. And when we talk about cannabis today, it's the high THC that is impacting on the developmental processes. And perhaps there may be other cannabinoids. And I don't think that CBD shouldn't necessarily be taken during pregnancy, but in terms of potentially its treatment for psychiatric disorders, and it's being looked at in many research studies as that might be something to look at. But the cannabinoids are not equal, and that's one thing we really need to emphasize. So this is my full team. We do a lot of work looking at prenatal effects of the drugs, and our clinical team that for us to see whether or not that's relevant to the human conditions. And like I said, my partner in crime, Yoko Nomura, who was we're masochistic in doing these studies. Dr. Sals's amazing addiction medicine doc, and all of the team that looks at, like I said, these developmental effects of cannabis and beyond. I will stop there and start taking questions.